a little bit confused. This is a report of a follow-up of a study we did in Milwaukee at the what was then the Aurora hospital system, and has now become Advocate Aurora. The work reports a follow-up to what's called an EPET-1, and that means an electrical property evaluation technology, where we studied the electrical properties of tissue that had been excised during breast cancer surgery. And the patients were consented and asked to participate with follow-up medical visits. It was then, which would then be recorded in the Aurora Cancer Registry. An average of about 8 years of follow-up data were acquired over a 10-year span. Reference 1 is the result of that study. The follow-up is a recently published brief report, and it is there's a link, and by the way, the link is free. So, as I said in Mad Magazine, for free take for buy waste time. So in this presentation, I'm going to review both of those studies because a lot of people will probably not be completely familiar with them, and then give you my take on a hypothesis to explain them. Okay, so the first question I have is, what is unique about tissue electrical properties as they're compared? Are they comparable to inanimate materials? And of course, the answer is no. They're not, and if you took a whole bunch of chemicals, put them in a pot, stuck some electrodes in them, and measured the electrical properties, they would not have any really relation to this, what you see with the tissues. Of course, the reason for that is, simple answer to this is the cell that is the difference. And we know what that is causing that difference. It's the cell membrane. This is a picture of the cell membrane, rather stylized, but showing a lot of stuff goes on in the cell membrane. Most importantly, the membrane itself is a double layer of lipids, so arranged that the interior side, that lipid is what they call water. It doesn't like to have water, but the two sides it do, and so they connect. And there's very little electrical conduction that goes on in there. The resistance is extremely high. However, the charges on that side, a positive charge would produce negative charges on the inside. And given that there's very little resistance here, if the resistance was a very high resistance, that would then make a rather good electrical capacitor. And so what do you do to make use such a measurement? What does it tell you about the cell? Well, first of all, what to do is you measure the voltage drop across the tissue divided by the current, and it's called the impedance. And you can see that if you have current coming in through the interstitial fluid or matrix, when it gets to the cell, it's either going to have to go through the membrane, through the capacitor, or going to have to go around. Okay, so you need to decide how to do that electrically. And this is a fairly simple circuit. Others have been proposed, but this one works well enough for this presentation. The tissue comes in from the other cells, and then when it gets to the cell, either it's going to go through the capacitor or it's going to go around. The capacitor will store the charge, but only so long as this resistance is very, very high. The characteristic time for it to relax, and that's what the term relaxation means, for the charges to get transferred back to the other side again. That characteristic time is this resistance times this capacitance. It's simple. So I'm going to now show some math, and having watched a little bit of this previous things, I think this is going to put some people to sleep. I'm sorry about that. I'm not going to do a lot on this. I just wanted to show you that there's a way of describing this. The first thing I want to tell you is we use the square root of -1, and I'm sorry to tell you, but if your primary school teachers were telling you you can't use the square root of a negative number, they lied to you. You can, and it produces two particular types of results. One is a so-called real result, and the other proportional to the square root of -1, which is interestingly enough called the imaginary component. This in the physics or electrical engineering world, this is the impedance, this is the resistive component, this is the reactive component. Now, the plots show what happens. This parameter alpha was introduced by the Cole brothers. That's why we call it the Cole relaxation frequency. And what it is it's just a dimensionless number. If that number is 1, it's exactly the description of the circuit I just showed you. But if it's less than 1, it changes. Now, here's how it changes. If it's at 1, the resistance, effective resistance simply drops to 0 at above the frequency, in this case, 50 kilohertz for the example. Why? Because the capacitance shorts out the parallel resistance. Back to this capacitance, the impedance of that is very, very high at low frequencies, but it gets proportional to the frequency, the conductance goes up to the point where it just simply beats out this resistor. So that is useful because then you can do something with it to measure something going on in the cell. However, you do have to have the ability to fit the data. And what the Coles found was that this parameter, which is actually a broadening parameter, if it's 0.5, for example, you get these purple curves. And on the reactive component side, what you get is it's really a negative number. You plot it with the times minus sign in order just to keep it all in the same, in an easy plot. But you notice something. The peak frequency, the place where this frequency is equal to the capacitance or the Hall function, that does not change with the width of the plot. So I'm going to go back a little bit. This is some of the early data we took. I think it was 18 years ago, and we first 25 or 30 some plots, I remember. And that is, I'm plotting the peak frequency versus essentially what is the width of the thing. I'm not using the Cole function here. I'm just plotting it. So you have red dots for cancer. Breast fat is a solid, is a rectangle or a square, and a benign triangle, green. You kind of look at that and you say, well, there's some groupings here, but it's particularly the benign. This doesn't seem to be real good. Until you rotate the graph. Now you see something that would have been hard to see before. Notice that all the reds are in this, essentially within these boundaries. One little stripe right here. The blue ones, the skin, or excuse me, the fat are here. And the green, the benign are here. So what it says is, as you increase the the relaxation frequency, then which is 1 over that time constant I talked to you about, that as you decrease that or increase that, you go from benign to cancer, and then this is just breast fat. So it says that there's looks to be quite a difference in the separation. it was the beginning of our realizing that this was a good classifier. And this is what we got when we used that as a classifier. You just the, you don't need to worry about the width. The width is generally scattered quite a bit because it has other features in it. And you don't need that width. You only need one parameter, the Cole frequency. Here's a plot using likelihood, the ratio of, in this case, red is cancer to ductal material. Ductal is a term at the time. This ductal is pure or high ductal content breast material sitting in the breast fat and the stroma. And what we found was the probability peaked and was very narrow. It was like 10 to the 5th hertz up to about 2 times 10 to the 6th for breast fat, but for cancer. But for the benign tissues, it peaked at about 2 times 10 to the 3rd. The difference between these two is 3 orders of magnitude, because this is a log plot. If we were sitting in an auditorium looking at this on a big screen, in a linear case, that peak would be somewhere out in the parking lot. So this then was firsthand really good evidence that what we have is a good classifier. We then use that same technology, the same method, likelihood ratios, to look at the patient's results compared to the Cole frequency at the time of the surgery. So we had new data. In this case, it was characterizations or the follow-up visits of the patients. And the results there fell into the following categories. Where there was cancer originally, if the original measurement was between 1 and 3 in this normalized set of data, if you go from 100 kilohertz to 2 megahertz, divided by 100 just produces a dimensionless number between 1 and 20. It's easier to work with. So we would use that. Between 1 and 3 in these normalized cases, there were no patients found that had a cancer that had recurred. Between 3 and 6, the cancer recurred, but there was no metastasis, because we call it RNA. And then the last group that was identified was cancer with metastasis, and that was anything above roughly 6. So that was one thing we did. And then we did another statistical analysis. This is the kind of thing here. It's shown. The green curve is what's they call an exceedance average. When you get data, you get what do they call it? Data that's been censored, and not the great censor in the sky. It's just data that's missing. So if you like with a newspaper, if you blanked out the little pieces, you would have stuff that's missing. If you take averages, typically averages are more useful in that case. And the exceedance is the average of all the data above a certain point. For example, if I were to go here on this normalized scale to, let's say, you had an original value of 4 for the Cole, normalized Cole frequency. The exceedance average would say that you had 4 more years. It's kind of interesting that was exactly 4, but at any rate. So if you had a 5, it would be about 3.5 years, and so on. More to go before in the case of an engineering guy, before the part you were looking for is going to break down. Okay, and that's where it's used in engineering. So notice that you get very sharp changes of slope between these different regions. So the boundary then between for example, non-recurrent and recur the p-value is about 10 to the minus 20. And similarly with RNMM to RM between the no metastasis and metastasis. So this is statistically significant, of course. What we've also plotted on this plot is the case where we have a Nottingham prognostic index. And you notice that that straight line is just all the same. So it's between the first and the second regions, similar to our regions. There's absolutely no difference. It's only when you get to almost metastasis, which may be just too damn late, that there is anything reasonably significant, less than 0.05. And as a result, we then decided to do a multivariate analysis. And this is using comparisons to, we used NPI and the grade, which is part of the NPI algorithm, and compared to other known receptor things like ER, HER2, and so on. And notice that the p-value for NCRF is much better than all the rest. So correcting for each of these, we still get much, and the red ones are the NCRF, get a much better result. This gives us confidence that we have been able to develop a classifier that really works. Now, the question is, where did this, what's going on here? You have to start considering the so-called field cancerization model. Slaughter suggested in 1953 that you don't get one isolated cell, but a whole organ is, and I think of it as the cells begin to change. They go into two groups, one that are daughters of the changed cell, and the others that are quite the same. But the amount of stuff that is changed and is cancerous grows as time goes on. So more recently, Harry Submarinian, and he's a student of Adam Backham. I think Harry must have been I think he was the principal author because he was his PhD thesis. And they measured the scattering of light in the cell. You can't make an image out of it, but you can measure the disorder of the cell. And just take a look at this one. This is the disorder function. This is the normal cancel as a control. Cell, not cancer, just normal. This is a tumor, very disordered. And even 4 centimeters away, it's disordered. And this is also seen in colon, pancreatic, and lung cancer. So a little working hypothesis to finish this up. We hypothesized that the long-term process of cells transferring from benign to cancer is connected to the relaxation frequency by the following. Transformation process produces disordered cellular material. We get that from Harry's work. The electropolarization, you can do even a back of the envelope calculation. I'm a physicist, would love to do that. We can show that the relaxation frequency will now change and increase from benign to cancerous, which then means that which we observed. And with Harry's work, it may also be a universal property of the cell. And this gives then the probable outcomes of the risk of metastasis for cancer surgery region can be predicted from knowledge of the original value of relaxation at the time of surgery. So you took the value at the time of surgery and you can predict what's going to happen up to about 8 years or so after. Okay, I don't know how the heck this got in here. This one's just a fun thing. I just going to say that to end, the result that I would like to see happen is we get a long-term study going that could look, let's say, have it working in the path lab or the histology lab. We spent a lot of time over the last 2 decades, and all the tissues that come in, you can consent the patients. You would consent them to allow their tissues to be examined, subject to the being available, the tissue not being damaged. Well, it isn't damaged, but there's enough tissue. So you could do a lot of different things like Harry did with these 3 different cell types, and show how far this goes into the cell structures and how well we do. Can we do this? Clearly, the data here supports the idea of doing it for at least breast, and I'd just like to see it extended. Dr. Prakash, I'm done.
That's the presentation from Professor William Gregory. So, any questions for Professor William Gregory? You can directly ask, or else you can give your questions in the chat box. Any questions? Any questions? Okay, it seems there is no questions for Dr. William Gregory. Thank you very much for joining us. So moving on to the next and the last presentation of today's session is from Dr. Chien-Fu Hung from Johns Hopkins University, USA. Dr. Hung will be speaking on annexin A5 as an immune checkpoint inhibitor and tumor harming molecule for cancer treatment. Dr. Hung?
Can you see my slide?
Yes.
So you're good, right?
Yes, I can see your slide and you can go ahead, please.
Okay. So I'm going to talk about the one of the platform technology using annexin A5 to as the user.
I'm sorry, your voice is not clear, Dr. Hung.
The voice?
Yes, now it seems.
It's a little bit better.
Yes, it's somewhat a little better.
Okay, so so before I talk about my technology, I'm going to kind of talk about a little bit basic the T cell immunology, and then I'm going to update to you the current status of the immunotherapy in clinically. So first of all, the intent of the immune cell to control cancer cell, there are different kind of cell able to kill the cancer cell. For example, the CD8 cytotoxic T cell, NK cell, leukocyte activation killer cell, or the macrophage that find the antibody and then kill the tumor cell called ADCC, or the macrophage can secrete the TNF, nitric oxide, or superoxide can kill the tumor cell. And I'm going to focus on CD8 T cell. The CD8 T cell is the most powerful killer cell. The reasons for good reason. Number one, the T cell, CD8 T cell, they can recognize the tumor antigen very specifically. Even one mutation of antigen, CD8 T cell are able to recognize. Secondly, that CD8 T cell, they kill the tumor cell very efficiently. one cell can kill one tumor cell, and after kill one tumor cell, they can continue to kill another tumor cell. It's like serious killer. And third reason is the CD8 T cell, when they recognize the tumor cell kill them, they are able to amplify and proliferate. One become two, two become four. Finally, the CD8 T cell, after kill the tumor cell and tumor cell go away, but sometimes tumor cell maybe come back again. And CD8 T cell have a memory function. They will still memorize the tumor cell and then continue to kill the tumor cell. That's why the CD8 T cell has been one of the key focus for the cancer immunotherapy. Currently, the clinical immunotherapy that have three categories. Number one is adapt transfer tumor specific T cell. Number two is therapeutic vaccine. Number three is the checkpoint inhibitor. So in terms of the adapt transfer tumor specific T cell, that you can do it simply is isolate the tumor specific T cell from the patient and in vitro amplify, expand it, and put back the patient to control tumor cell. Or what you can do is that the slide show you can do the genetic engineering the tumor specific T cell. What you do is you isolate the T cell that they can recognize the tumor specifically, and you clone the T cell receptor recognize tumor antigen, and you put in the viral vector, and you engineering the CD8 T cell, and then you expand it, and then put it in the patient to control tumor cell. Or you can clone the antibody, recognize the tumor antigen, and use the FAB region and fusion with the CD3 zeta chain or other molecule, and clone the viral vector, and engineer into the CD8 T cell and infuse into the cancer patient into control tumor cell. And adult cancer therapy has been very, very successful for the liquid tumor. And second category is the therapeutic vaccine. Therapeutic vaccine currently in US, FDA only approved one vaccine, the dendritic cell vaccine for the prostate cancer. So the way it works is that you isolate the monocyte from the patient, and you culture with the FAP. PAP is a protein-specific antigen fusion with the GM-CSF, right? So PAP is a tumor antigen. GM-CSF are able to mature the dendritic cell, and you infuse to patient. Once you infuse the patient, what happens is that the antigen uptake by the dendritic cell and GM-CSF mature dendritic cell. So that will lead to able to activate CD4 helper cell or CD8 T cell and then to control tumor. Okay, so that's the second category of the therapeutic vaccine. And then third category is the checkpoint inhibitor. The checkpoint inhibitor is being worked very well in a lot of cancer, right? So there are two categories of the checkpoint inhibitor. One's the antibody against CTLA-4 or PD-1 or PD-L1. So what happens is that when dendritic cell activate the T cell, and CD8 cell will upregulate the CTLA-4. And what happens is CTLA-4 will turn off the CD8 T cell, downregulate a lot of molecule on the CD8 T cell. So if you can use antibody block the interaction of the CTLA-4, and then the CD8 cell can continue to activate. Another one is the PD-1 and PD-L1 interaction. The dendritic cell activates CD8 T cell, secrete a lot of cytokine, and lead to upregulation of the PD-1 or PD-L1, the program cell death 1 and 2, and interact with PD-1 on the T cell, and lead to the T cell apoptosis or tolerance the T cell. So if you can use antibody block PD-1 or PD-L1 or PD-L2, then you're able to continue activate T cell. That's the second way to kind of lead to activate T cell activation. So currently the response rate for this kind of checkpoint inhibitor around 8 to 44%, and many of these kind of response are very durable. However, majority of the patients fail to respond to immune checkpoint monotherapy. So people in the field continue based on the basic immunology. And here's the kind of the long list of the immunomodulator molecule or checkpoint inhibitor. the people continue development, right? So the purpose of this is now try to, let you look at the detail of each molecule, just let you know there are so many molecule people continue to develop. So in our lab, we focus on one particular checkpoint inhibitor called enzyme, and the reason I'm going to tell you the following. So enzyme are able to bind in the phosphatidase serine called PS. So what happens is the PS is a very powerful inhibitor of the inhibit polarized M2 to M1, the macrophage. Also inhibit the T cell modulation, inhibit T cell activation, inhibit the effector function. So it's a very powerful inhibitor for all immune cells, right? So if enzyme can block the PS and enzyme can serve the global checkpoint inhibitor in all the immune cells. So the next thing is that what was enzyme? Well, most people know enzyme is the assay for apoptosis, right? So what happened is that normally the PS is inside the cell. Once the cell become apoptosis, PS will display on the surface of the cell, right? So it's apoptosis assay. Enzyme has been served as apoptosis assay. So what happened clinically, people has been published in Lancet or Nature Medicine. What they did is they use an enzyme conjugate with the radiation agent and put into the patient to look into apoptosis of the tumor cell after the chemotherapy. So an enzyme has been used clinically and it's very, very safe. Okay. So the PS, right, is also can serve as the universal tumor target antigen. The reason is because that usually the PS inside the cell not expressed on surface, but what happened in the tumor endothelial cell, they are dispersed on surface of tumor endothelial cell. So what it means is that potentially the PS can serve as universal tumor target antigen. Okay. Then we talk about the PS, we talk about apoptosis. And one thing to keep in mind is apoptosis very, very powerful. They induce immune tolerance. The reason they can induce immune tolerance because macrophage will uptake those apoptosis cells and lead to secretion of anti-inflammatory cytokine like TGF-beta. And TGF-beta is most powerful immunosuppressed cytokine to suppress T cell function. Okay. Besides that, naturally you have life and death of the cell in our body. Another thing we know that in terms of the cancer treatment, we use a lot of chemoradiation. So what happens is the tumor cell, when we treat the chemoradiation and cell will die and then uptake by macrophage and secrete a lot of stuff. And those kind of stuff not only suppress the T cell and those kind of like TGF-beta also can facilitate the tumor cell continue to proliferate. So it's a terrible situation when you treat the chemotherapy, potentially lead to immunosuppression because the TGF-beta potentially also lead to the continued expansion of the tumor cell because those cytokine. That's the dark side of the the chemo radiation treatment. But the bright side is that people also know that when you treat the chemo radiation, what happens is the tumor cell die. And the two things happen. One is the tumor cell release tumor-specific antigen. At the same time, also release a lot of immunomodulator molecules like HISHA protein, calreticulin, release DNA, release RNA. It can serve as an adjuvant, right? So here, tumor antigen, you have those immunomodulator molecule release. And what happened that the T cell will obtain those antigen, mature, will educate T cell, and then make the T cell function very well and to control tumor cell. So that's the good thing in terms of the chemoradiation, right? So you deal with two pathways. One is that potentially they will be helpful after chemoradiation that reduce a lot of good immunomodulator molecule, right? Activate T cell. Although the other way, apoptosis cell, because the PS uptake by macrophage is immunosuppression. So you have two pathways, right? So enzyme is the answer that will only push only one pathway rather than two pathways, right? So enzyme can bind in the PS on the tumor cell, right? And then avoid the uptake by macrophage, and then will lead to the anti-immunity rather than the immunosuppression, okay? And here's a readout showing that the case that NAC5 inhibited TGA-beta production by the antigen-presenting cell. As you can see here, right, when you have antigen-presenting cell, when you have tumor cell called TC1, this is cervical cancer model that express the human papilloma E6, E7. And then when you choose the cisplatin, you can see a lot of TGA-beta secretion. When you NAC5, then it will block the TGA-beta secretion. And here, this example shows you that, PS is very powerful. The immunosuppressive factor suppress the T cell function to secrete one of the cytokine. Here's an example, interferon gamma, right? So once you activate T cell, they will secrete a lot of, interferon gamma, but NAC5 doesn't change anything in terms of the activation of T cell status to secrete interferon gamma. But when you add a PS, they'll completely shut down the interferon gamma secretion. When you add the NAC5, able to reverse this kind of suppressed factor, so T cell continue to secrete the, interferon gamma. So that's what it means is that NAC5 can block the PS and then lead to reverse suppressed function of the PS. Okay. And here's the slide to show you that NAC5 able to target the tumor location. Okay. And the way we do is we use NAC5 fusion with the Gaussian luciferase. Okay, and this is tumor mice. You inject PS, you inject Gaussian luciferase, or NAC5 Gaussian luciferase. Okay, so you can see that a little bit signal that increase in tumor location. So this is luminescent signal in the tumor, right? When you inject NAC5 Gaussian luciferase. And here, the bottom here is that when you take the tumor and treat with the cisplatin and followed by inject those reagents, you can see the signal of the luciferase further enhanced. So what I mean is that NAC5 are able to reach the tumor, but once you treat with the cisplatin, because there's more apoptosis cell in the tumor, and then NAC5 will even further enhance the target tumor location. And here's a slide show you in vivo that the tumor-bearing mice, you treat with the chemotherapy cisplatin followed by NAC5 injection, and to look at the tumor microenvironment. And for example, look at the TGA-beta again, right? So you have the tumor-bearing mice treated with the cisplatin. You can see a lot of TGA-beta. When you inject the NAC5, it will suppress the TGA-beta secretion. We also look into the immunosuppressor cell, like T-regular T cell, the cell that inhibit the CD8 T cell. We also look in the minority suppressor cell. They also suppress the CD8 and CD4 cell. As you can appreciate, when you choose the cisplatin, you have a lot of Treg. When you inject the cisplatin plus NAC5, that will suppress the Treg number. Here, show you that the NAC5 able to suppress the myelosuppressor cell. We also look at the bright side of this, CD8 killer cell or CD4 helper cell, right? So when you choose the cisplatin plus NAC5, they were able to increase number of the CD8 T cell and number of the CD4 helper cell, right? So what I mean is that in the tumor-bearing mice after chemo treatment, they're able to decrease the factor, increase the good, cell that in the tumor, right? And then most importantly, we're going to look at the anti-tumor effect in vivo, right? We're going to take the TC1. Again, this is the cervical cancer model. Tumor cell express the human papillomavirus, the E6, E7 antigen, and we choose the cisplatin, and followed by inject the HPV E7 long peptide intratumorally, and followed by inject NAC5. And